All right, thank you very much, Marty. I need to mention, in addition to being part of the School of Forest Resources and Conservation, I'm also affiliated with the Florida Aquarium, who house me and help provide field support. I'm housed down in Hillsborough County. So I want to change track a little bit from production aquaculture and switch into aquaculture for active restoration of corals in Florida, which is something that's really been taking off in recent years. In terms of relevance, most of you are probably pretty familiar with the importance of coral reef ecosystems. I don't need to harp on it too much. This is from a series of photos by Phil Dustan, which many of you have probably seen. But I like to use this one in particular because it shows the two coral species that we primarily aquaculture in Florida right now. We're in the process of developing others, but the two primary ones are staghorn coral, which is down in the front left of the photograph, and then elkhorn coral, a congener, which is in the background and on the right of the photograph. And this photograph does a good job of showing you sort of the spatial dominance of these species historically. When you talk to people that dove in the Keys in the 60s, you had to swim through the acroporid corals to get to the interesting corals. So they were just really dominant. And this is the same site in Jamaica, a more current day photograph. And the factors leading to this decline are numerous. Another Joshua Voss, I think, is going to talk to you later this afternoon, probably in a lot better detail than I could about some of those factors. But we've got land-based sources of pollution, we've got climate change, ocean acidification, and a host of other global as well as regional factors that have led to these declines. This is a Florida relevant picture. So this is Carey's Fort Reef in the upper Florida Keys. This is primarily a pomata zone, historically a little bit shallower coral, but you can see the degradation across time to where today what we have is essentially an algal-dominated rubble field. The other factor leading to this decline that I need to mention is the die-off of a keystone herbivore on reefs in the Florida Keys and throughout the Caribbean. So the long-spined sea urchin historically provided control of algal growth on reefs. And in the early 80s, there was a big die-off of those. It's still sort of unexplained. And so it led to a lot of these declines. In terms of economic relevance, natural reefs, so these figures are only for natural reefs, and these are estimates provided by NOAA, but 44,000 jobs roughly. And that asset value is derived from tourism and recreation as well as shoreline protection. So having this big geological structure relatively near shore provides a lot of abatement of wave energy and that kind of thing. What these figures don't account for is contributions to fisheries production. So we've heard a lot about spiny lobsters and then some of the fin fisheries in the Keys that are so economically important that are a little harder to quantify, and so are not included in these figures, but are no less important, certainly. So these two coral species were the first corals listed on the Endangered Species Act. They're listed as threatened, and they were listed in 2006. Part of being listed as an endangered species means that you have to come up with a recovery plan. So NOAA was tasked with coming up with this recovery plan, which came out in 2015. It includes, in a big way, the threat-based criteria that I talked about. So doing something about climate change, carbon emissions, land-based sources of pollution, that certainly needs to be addressed. We definitely understand that we are not going to restore or outplant our way out of the problems that are facing reefs globally and especially in the Caribbean. However, this recovery plan does also include population-based criteria, so using aquaculture to generate coral biomass that wouldn't otherwise be there. And then also the other point to make is that any of the acroporid corals that are in nurseries in Florida are tracked, they've been identified, their genetic identity is known using molecular markers, and they're tracked throughout the production process. So we have documented, other scientists, not myself, have documented that having these corals in nurseries has preserved genetic diversity that would have otherwise been lost due to mortality events. So while active restoration does appeal to that part of our human nature that wants to do something right now and make a difference and not be happy with delayed gratification, it does have other virtues in terms of the things I just mentioned. I use this figure a lot. I stole it from Allison Molding from NOAA. I really appreciate Allison letting me use it. It shows the spatial scale of Acropora outplants throughout the Florida Reef Tract. So from Broward County all the way down to the Dry Tortugas, there have been significant outplantings of these corals. Whoops, don't want to skip that one. That's an important one. These are all the partners that operate ocean-based nurseries in Florida. So you've got Dave Gilliam's group at Nova Southeastern, Diego Lerman's lab at Miami, the three nonprofits, the Coral Restoration Foundation, Mote Marine, and also the Nature Conservancy, as well as FWC, a state agency. And these logos roughly correspond to the geographical location of the main nurseries for these facilities. In terms of impacts from the hurricane, news is sort of still starting to trickle in, but one anecdote that I will point out that I heard in an email chain was that a tree, one of the structures that they grow the corals on from Moat's nursery down around Summerlin Key, actually washed up after the hurricane in Broward County, up by Nova Southeastern, with live corals on it. And so the Nova folks were able to actually take those corals in, and they're in the process of stabilizing them. So that was just sort of an interesting anecdote that shows you, one, sort of how resilient they are, but two, how much everything got washed around by the storm to such a huge extent. This is the numbers of outplanted coral. We're now at well over 100,000 outplanted corals in Florida alone. If we had, again, thanks to Allison Molding for this figure, if we had 2016 data on here, I would guess that it has leveled off quite a bit just based on some of the big outplanters and how much they outplanted last year. And the bottleneck that we're seeing is not in terms of production in the nursery, it's in terms of outplanting. So we can grow corals in the nursery way faster than we can physically take them back out to the reefs at this point. So there's a lot of interest in developing techniques to increase the efficiency of our outplanting. The other thing to note is that we get really good survival for a couple of years. It's not uncommon to have like 90% survival after a year, 80% after 2 years. And we haven't really been doing it long enough to document, but we sort of are having an inkling that after a couple of years, that survival is declining, as you might expect. But we do still have some of these original outplanted corals that are out there and producing fragments and surviving in places that otherwise didn't have coral. So moving from sort of the meta scale, what's going on with coral restoration aquaculture, I want to talk specifically about research that we've been doing to answer questions. I've been fortunate enough to have a couple of really terrific grad students that were focused on coral. Katie Lohr on the left is a PhD student that started in 2015, so she'll finish up in the next year and a half. And then Kelly O'Donnell was a master's student. It's an Aylesworth scholar, so thank you very much to Aylesworth family and Florida Sea Grant for that opportunity for Kelly. Kelly finished last spring, and she's now working for NOAA Sustainable Fisheries in St. Pete. I want to start off talking about, oh, here they are again, sorry. In the water, Katie on the left, that is not a Florida reef. That's the Great Barrier Reef. That's a project that we did in Australia this spring. And then there's Kelly monitoring some of her outplants with Moat. So I want to talk about the first half of Kelly's thesis because I think it's a good introduction to the production methods that are used to grow coral. So Moat was a great partner in this study. They pretty much let Kelly come along with them and funded all of the field work for this in kind. And this study was designed specifically to answer questions that Moat had. Panel A is one type of benthic attached culture method. So we were really just directly comparing two culture methods for corals because we had an idea that the tree method, which was developed by the Coral Restoration Foundation as a version of a water column suspended method, could produce coral a lot more quickly than these benthic attached methods. If you think about it, that's a really unnatural way for a coral to be in the ocean, just kind of suspended and free floating around. But we knew that it grew faster, but we didn't know how much faster. And Moat was also interested in being able to quantify the difference in these two methods and estimate production, so shifting production back and forth between these methods based on their needs. And we also had some questions about potential trade-offs between these two growing methods in terms of downstream outcomes with outplanting. So Kelly went out once a month for 12 months and collected data in Moat's nursery. The solid lines, oh, the other thing I need to mention, we weren't really interested in genotype with this study. We were just looking at the production methods, but we know that there are differences between the genotypes, so we needed to control for that. So we used sort of randomly selected 4 genotypes that were available. The solid lines are trees or the suspended method, and the dotted lines are blocks or the benthic attached method. And what we found was that even accounting for breakage, so 1 in 5 corals that we put out on blocks broke during the study. That doesn't mean they died. That's pretty much a natural process. But we didn't have any breakage on the trees. Even accounting for breakage, the trees have an intrinsic growth rate that's 2.5 times faster than blocks. And so the other interesting thing to note is that the post hoc rankings on these genotypes were identical. So the fastest was the fastest regardless of growing method, and the slowest was the slowest. So that gave us some hope that there might be some sort of consistency in terms of the phenotypic characteristics of these genotypes. The second half of Kelly's thesis was going to be taking these exact corals from this experiment and outplanting them side by side to see if there were differences in outcomes based on growing method, because we sort of suspected that there were. Unfortunately, the dotted line here is temperature. 31 degrees is a thermal threshold for the species. And we had more than a month of daily mean temperatures that were above 31 degrees. So pretty much all but 2 of the 120 corals in this experiment died by the end of the study. But we ended up just outplanting the same genotypes. I don't have time to talk about that, but there are some interesting findings from that as well, and we're still in the publication process. This study was done in Little Cayman with the Central Caribbean Marine Institute. Katie Lohr did this, I want to say the summer of 2015. It's not done in Florida, but it has relevance for Florida because the reefs are really similar in terms of species anyway. In Little Cayman, they have these 3 distinct reef zones that are all within 400 meters of shore. So you have the shallow back reef area, zone A. Zone B is sort of like an intermediate reef slope, and that is actually where the nursery was located in this particular study, and it was a water column suspended nursery. And then zone C is sort of your classic spur and groove, 40 to 50 feet staghorn coral habitat. Before we started this study, we actually did roving diver surveys and determined that there were remnant populations of staghorn coral in all 3 of these zones. So it's not like, so all 3 of these zones support staghorn coral, which we thought was good justification for trying to restore them. So what happened when we took corals from the nursery and outplanted them to these 3 zones, in zone A, we had decent survival, but a ton of breakage. So that's what the corals looked like relatively soon after being put out. There would be a healthy staghorn coral directly adjacent that was totally fine, but something about the nursery production method was it appeared to be leading to some sort of breakage when they're outplanted in a high energy environment. In zone B, they did really well, good growth and survival. And then in zone C, we got about 40% mortality really quickly right after outplanting, which is really unusual. So taking them from a shallower area to a deeper area for some reason led to some mortality. And that's part of what that Australia study was looking at. We're working with some photophysiologists to see what light and some other factors might be doing. So the take home from this is sort of that if you want to restore different reef zones, you may need to think about different ways of constructing your nurseries or maybe acclimating your corals as you're moving them around. Because again, it's fairly unnatural to take a coral from one zone and move it really quickly and glue it down somewhere else. Ooh, okay. Last study. This one was with CRF, another study that Katie did. Basically a big common garden study looking at phenotypic variability among these genotypes. This one we actually brought them to shore and weighed them too, which was a huge pain in the butt, but it gave us this a really neat insight in terms of some of these corals that seem like they grow really, really fast on the trees actually weigh almost the same as corals that are much smaller. So there's a trade-off there in terms of mass per unit length on the coral that we need to think about. And the other thing we documented with this study was some intraspecific variability in thermal tolerance. We were lucky enough to have a non-lethal bleaching event towards the end of this study, and so we documented genotypes that were less resilient, more resilient to bleaching, and everything else sort of fell out in between. So this is something really important moving forward because thermal stress causes a lot of problems. In conclusions, the genetic diversity is something we need to keep working on. There are a lot of intricacies there to look at. It's not going to be as straightforward as identifying really resilient corals. Outplant research needs to be done a lot more. It's sort of in its infancy. The Coral Restoration Foundation actually just got a big NOAA grant to outplant some of those genotypes that we looked at as well as others and study them in more detail. And then collaboration, I have to point out, NOAA has started a Coral Restoration Consortium where we get all of the partners together, exchange ideas and information, and make sure that we're all working towards a common goal. With that, thanks to some of the other funding that I didn't mention and FKNMS for permitting.
Great.